everyone, and welcome to the Ecological Continuity Trust webinar series. A little bit of housekeeping before we start, just a reminder to keep yourselves on mute, and if you wouldn't mind turning off your video, that would be great. If you have any questions, do feel free to pop them in the chat function. There's a little speech bubble in the bottom right hand corner. You can pop them in there and we'll pick them up at the end. So please do pop things in the chat as you think about them, and we'll be looking forward to having a discussion at the end. I'm very pleased to introduce to you Professor Rob MacKenzie, who's Professor of Atmospheric Science at the University of Birmingham and Director of the Birmingham Institute of Forest Research. Rob studied chemistry in Edinburgh before doing a PhD in computer modelling of air pollution at the University of Essex, and this is where he first encountered the profound effect that trees can have on the atmosphere. After that, he spent 10 years at Cambridge working on the ozone layer before taking up his first academic position at Lancaster University. In 2021, he moved from what he describes as the rainy northwest, obviously being in Lancaster, I will confirm that, to the eternally sunny West Midlands, which I think is a little optimistic. I'm a Brummie, so I've come the other way. And he's now has his chair at Birmingham. So without further ado, I will hand over to you. Thank you.
Thank you very, very much, Carly. I thought you might have a different perspective on the climatological differences. And I think I probably typed I mistyped when I was sending my biography details. I've been in Birmingham for I think it'll be 11 years this summer. And to say it's, hopefully that's come through to you all, just to say what pleasure it is to be able to present at the ECT today. And I'm going to give you an update, because I did speak at an ECT meeting in 2019, and this will be an update from the BIFoR-FACE experiment, which uses elevated CO₂ as its experimental treatment. And my screen is slightly obscured, but I hope you could see a subtitle which says half-time first leg. So it's definitely half-time in the sense that we've been through 5 growing seasons out of 10. It says first leg with question marks after it, because we've got absolutely no indication that we can take this experiment into the second decade. But what a lovely thing that would be if we could. Just to give you a little bit of nomenclature clarification. So the institute, BIFoR, is a very broad-based institute, and it acts as an umbrella virtually across the University of Birmingham, providing a place where fundamental science, social science and cultural research related to what we call forested landscapes. So any landscape in which trees are an important part anywhere in the world. And there are quite a lot of elements to the institute, therefore. The one I'm going to talk to you about today is the BIFoR-FACE facility, if you like, our keystone facility, which is very much focused on the impact of climate change on the productivity and resilience of temperate forests. But we're also very interested in pathology, plant pathology, and the resilience and resistance of trees to invasive pests and diseases. That is something that has become much more prominent in the Institute just over the last few years, I'm very pleased to say. We're interested in the value question, so these questions of the importance of forests to humans, but also to non-humans. There's a big sea change happening in social science and environmental humanities, which is really beginning to take the non-human actor into account. And then we've got an education program which is actually moving forward quite strongly now, which is not about delivering forestry degrees per se, but it's about making sure that those people who are coming through our other degrees in geography, environmental science, biosciences, and indeed some others, are well placed to move into the forest sector, because we really think we need that breadth of skills moving into the sector. I'm not going to spend a long time giving you the rationale for why we're focusing on temperate forests, or why we're doing the experiment, but just a couple of things. We're interested in the global carbon cycle, particularly how that carbon cycle is being perturbed by fossil fuel, predominantly fossil fuel emissions and land use change. And forests play a very big part in that carbon, in the response to that carbon perturbation. And currently, the best estimates are that Northern Hemisphere forests absorb about 3 billion tonnes of CO2, of which the temperate forest, which is the green bit in the picture that's on the top of that slide over here, accounts for about 0.8 billion tonnes of CO2 absorption. So that's about the same absorption as the EU 27 emissions. That's the EU when the UK was inside it, so it's 26 plus UK. Or you could think of it as balancing the emissions from global deforestation. So the temperate forest is a really significant player in this global carbon cycle. That's the case now. The question we're interested in, particularly, is how things will change through the next century, let's say. We often ask people to focus around the middle of the 21st century. And this is the rationale for that. This is getting a bit old now, this slide, in that it refers to the IPCC report before the current one. But it uses this productivity gain parameter. So this is the amount of productivity in terms of kilograms of carbon per meter of latitude band per part per million increase of CO2. So it's about the feedback of CO2 onto that absorption into the land. So it's a feedback parameter or a gain. And all I want you to notice is that it has been estimated using the dynamic vegetation models. They have a central estimate, which is the darker line, and then this green band here is the uncertainty. And of course, it's large and uncertain in the tropics, and if time permits, I might say a word or two about the tropics towards the end. But it's also very uncertain in the temperate regions, and so there really is a pressing need for us to understand what's going on in the Northern Hemisphere temperate forests. There are lots of ways that you can look at how the land carbon sink, the absorption of carbon dioxide by the land, is going to be affected as environmental parameters change. But ultimately, so you can do these things in the lab, you can do these things in glasshouses and growth chambers, common garden experiments. They're all really important, and they focus in on important elements of the story. And then, of course, satellite and other kinds of Earth observation are very important for telling you what's happening now and looking backwards, seeing how things have changed over recent time as an indicator of how things might change going forwards. And there's been some brilliant work around the change of the seasonal amplitude of the seasonal cycle that is a good example of that. But if you really want to test directly the impact of changing global environmental parameters on the environment, you have to take the laboratory into the environment, as we all know in ECT, because that's what our long-term experiments are generally doing. So I don't think I need to belabor that point, but what we're doing is looking at the forest, so as a system, as an ecosystem. That's why we're doing it outside. That's why we're doing it in an established forest where the above ground and below ground are very much used to each other. They've been in contact for a long time, very closely interconnected. And we're changing just that one parameter, carbon dioxide, because carbon dioxide is increasing very rapidly and is continuing to accelerate alarmingly, even in spite of Kyoto, in spite of Paris, in spite of Glasgow. We haven't even begun to slow down, let alone reach a maximum and turn over. But our understanding is if the Paris Agreement does gain some really serious political and social traction, that by about the middle of the 21st century, about the middle of this century, we will begin to see the maximum CO2 in the atmosphere. And and that may well be about 40%. If we're lucky, it'll be about 40% above where we are now. If we're unlucky, it'll be even much higher than that. And that's what we're doing. We're adding about 40% to the current CO2, ambient CO2 in our experimental treatment. Just to be a bit more precise, we're adding 150 parts per million above whatever the ambient is that we're looking at day to day. And the way to think of that is that we're dropping the forest into the atmospheric composition of the mid-21st century. We're not changing the temperature or the precipitation to what we might forecast will be prevalent in the mid-21st century. Love to do that, but then our £50 million project would become a £150 million project or even more, because we would need to massively expand the infrastructure and the treatments. We started thinking about this in 2014. We started building it right at the beginning of 2015. We have some data from 2015 and 2016 growing seasons, which we can use as baseline. And then the treatment is currently designed for 2017, the beginning of 2017, the growing season of 2017 to the end of the growing season, 2026, actually. There's a typo in my slide, which would be 10 growing seasons. And we think we need to have 10 growing seasons because things change. And again, I don't think I need to belabor the point in this community. Things change, as you know very well, in UK circumstances from one summer season to the next, one growing season to the next. There's only currently one other BIFoRFACE facility in the world. It's just outside Sydney, and we work very closely with that facility, BIFoRFACE, and have learned a lot from them, a very great deal. There's another facility called Spruce, which is in Minnesota, which uses open-topped chambers. So it encloses it's working at the forest, at the northern edge of the forest. So it's uses open-top chambers for its system, but it's similarly large to ours. And what else do I need to tell you about BIFoRFACE? I'll say at the end that it involves very many people already, and it is always open for people to get involved. So please do get in touch with us if you want to work with us. It does work, which is amazing when you think about these pieces of infrastructure. These are sometimes called arrays or patches or rings. So I'm pointing at them on the slide just now with a little laser pointer. The ones with the orange circle over the top, that's just a virtual orange circle there, just to show that's where the treatment has been applied. So on that one, and that one, they're getting extra CO2, 150 parts per million extra, and the ones in the middle are not. They're just getting fresh air blown at them. And the trace of the control and treatment CO2 is given for 3 years. At the top here, these are daily averages, and you can see that the treatment and control are very well separated. We hit the control and stay on it very tightly, and there's very little cross-contamination into the control treatment. And that's because most of the mixing is in the vertical. The aerodynamics around the forest are such that there is very strongly upward directed mixing. I've said here that the residence time of CO2 in the plots is around 3 minutes. It can be even less than that, which is why we need many tons per day of CO2 to maintain our treatment. There's a paper by Chris Hart from 2019 in global change biology, which talks about the infrastructure and how it's performing. And Chris is on the call today, so he may be able to answer some questions if I find myself tripping over. But what I would also like to do is just shout out a really, really big public thank you to Chris as the leader of an operations team that have kept this thing going in the face of incredible headwinds. It's a difficult, you can see, it's a difficult facility to operate just by its scale, even in good times. But when you've got a pandemic to consider, and then wartime to take into account, which has completely upended our CO2 supplies and the costs of CO2 and energy, it is absolutely a testament to their dedication that we have been able to work through this, even continuing scientific work through the depths of the pandemic. And I really want to pay a public note of thanks to Chris for his efforts. So let me just jump on. I hope I haven't skipped too quickly past all the details why we're doing things. I realize I'm already 15 minutes into my talk. I'm not going to say a lot about research questions. I hope you'll take it on trust that for all the data I'm about to show you, there are specific research questions. The facility as a whole is looking at the response of the forest as a forest. So how the whole system responds to that single parameter perturbation. What we're particularly interested in are carbon and nutrient fluxes and reservoirs, in as much as we can see them, measure them, and see them changing. And of course, the implications of those changes that we see. Well, the first thing to say is that measurements made at the top of the canopy in the oak canopy show that there is increased assimilation of carbon dioxide through photosynthesis if extra carbon dioxide is offered. So we're offering about 40%, a little bit less than 40% extra CO2. And on a bright, sunny West Midlands day, we see a maximum effect of about 33% extra CO2 going into the treatment patches compared to the control. That's not the average that we see over our growing seasons, because every day is not the very brightest day. And so overall, we see a change of about 23%. And this rather, it's an elegant diagram, but it doesn't come across very clearly in PowerPoint, I must admit. It's just showing how the strength of the effect, which you can look at by comparing blue and red here, or by looking at how these dots for 2018 and 2019 move away from the unit line as light intensity increases. Then when we take a view over all these days, we see an average of about 23% extra CO2 going into the system. Now, extra CO2, 23%, it's much less than the 40% we're offering, but it's still a very significant change to the carbon input into this system. If your calorific input was going up by a fifth or almost a quarter, I think you would notice it, and things would happen in terms of your physiology, and things are certainly happening in terms of the physiology that we're observing in the forest. We are seeing stem growth. So the carbon's coming in. Of course, the very next question is, well, where are the oaks allocating that carbon? And we are now seeing that some of the carbon is being allocated into stem growth, into woody stem production. That wasn't absolutely simple to see, because the individual patches are themselves inherently variable, and we have to take that into account in order to see the treatment effect. And ultimately, that meant we had to do some coring of the trees and dendrochronology. So it took us we had to go back a couple of steps before we could go forward. But what we're seeing at the moment is a really rather substantial increase in stem increment from the in the treatment patches. I should just say these are all, well, the stuff I showed you from Anna Gardner's work is published, so you know that is what it is. That's our best guess. This is very much still being pressure tested to make sure we haven't goofed in any way, shape, or form. But this is our current understanding of where we are. The other place that we saw carbon allocation, in fact, very rapidly and quite strongly, was below ground. The first line of evidence I can show you here is some, at the top of our slide here, just there's a single time point last summer. We dug down to 1 meter. It actually turns out, I don't know if anyone does this, anyone on the call does this routinely in their particular setting, but in our setting, getting to 1 meter without bringing in heavy kit is really arduous and really difficult. So very well done to the researchers who managed to do this. But we're seeing, so now in most of the slides that I show you from now on, orange is going to be elevated CO2 and blue is going to be control. And we see in the O horizon, the organic rich horizon, the A horizon, the B horizon, we see changes, well, not so much in this B horizon, for these finest roots, less than 1 millimeter, going all the way down to 1 meter. And I'm sure if we could get further down, we would see changes even further down, but that really might have to wait until some very destructive sampling at the end of the whole experiment. Even in the slightly bigger roots, 1 millimeter to 2 millimeter, we still see these big changes. Now, this is only a single time point, so I'm not betting the house on this, but just to give you some indication that there are changes that we're observing. When we look at 5-year averages for just 30 centimeter OA and B horizon changes, we do see big changes, positive changes in fine root production, both less than 1 millimeter and 1 to 2 millimeters, particularly in the O horizon, but also in the other horizon. Maybe not. Maybe something is. Maybe the A horizon is just staying where it is, and things are happening in O and B, and we need to just keep making the measurements to try and see if that is indeed what's happening. That would be an interesting sort of bifurcation if we were to be able to be completely confident about that. I wanted to show off what I think are some lovely experiments on root exudation carried out by Samuela and his team using these root boxes where they have perspex windows into the soil. They can do photography to look at root growth and hyphal colonization, but also they can get in there and isolate some roots. Eventually they have to take the root away and then scan it and classify it, which is what this image is it's taken from. And then but before they do that, they look on site at root exudates. So very carefully putting the roots into a solution and measuring the difference in the carbohydrate and amino acid over time in that solution. These first results are very much thanks to the dedication of Michaela Ray here, who got these results out even during the pandemic. And again, it's a single time point, but we have got some more work on this coming soon. And again, seeing excess exudate or greater exudate in the roots under elevated CO2, with perhaps, and here I must admit I'm by far not an expert, so an interesting hint that there might be a seasonal cycle here, and if there is, obviously we'd like to know more about that. Longer term measurements looking at soil efflux or soil respiration as it's often known, using these little robots. Initially we saw a very strong treatment effect under elevated CO2, but that seems to be going away. We're not absolutely sure. We've hammered these very latest data, cleaned them to the utmost degree, so we're still making absolutely sure that these data are believable, but we think there may be a declining treatment effect here. And if there is, we are definitely going to have to go back to some of our other measurements, including our carbon assimilation measurements, and make sure that if we, whether we are still seeing a strong treatment effect in them. So this might be a little bit of an early indication that things are beginning to change and the system is beginning to bring things back into balance or into a different balance. Over the 6 years that we've got data of soil available ammonium nitrate and phosphate, we see some quite strong changes. I've just put these blue bars in to remind you and remind me that there is a pre-treatment or baseline measurement here, and they're not equal, the numbers from the patches that were at that point going to receive elevated CO2 but had not yet received it. So of course these differences have to be factored into our understanding of the significance of differences that we see later. And that's true of almost every measurement that we make. And it's a measure of the heterogeneity, the challenge of working in this very heterogeneous system. So summarizing, we've got in the below ground part of the story, first of all, there are a number of lines of evidence actually that we've got 30 to 40% increase in root biomass. So we are seeing a strong allocation of extra carbon below ground into fine root production. That is leading to an increase in exudates, we think, given Michaela's work, and which will be repeated. And that then is stimulating changes in microbial carbon, nitrogen, and phosphorus, all of which have increased in our measurements. And that's leading soil available nitrate to go down quite strongly. Soil available ammonium, is actually increased somewhat in the treatment patches. And we have an initially quite strong, but subsequently decaying, appears rather strongly decaying soil efflux, soil respiration, which just rudely taking the average over all the years we have would still be slightly positive, 8% positive. So trying to feed that into a broader understanding, we've looked at the below ground responses, we've seen that stem response, the assimilation response. We're looking hard at whether there's a litter response or not. It's not jumping out at us, but we do need to look quite hard. We do know we have a change in the specific leaf area or leaf mass per unit area, but whether that translates into a gross change in litter, we're still working through. And our water budget still remains with lots and lots of question marks attached to it. Which, again, it's not jumping out that there is a change from the treatment. And of course, what we want to do with this, as well as just tell the story in the way I've just told it, is to push the data underlying these effects through to use them to challenge Earth system models, and particularly the Joules model. And that work is ongoing in Exeter, even as we speak. I just want to finish by very briefly telling you about access and collaboration, because I do want to make the point that the facility is open. It's an international facility, as I'll show you, as I'll emphasize just in a second. We've got something like 80 projects running so far, documented through project forms, which are subject to a reasonably light touch process, just to make sure that we're not saying yes to the same science more than once, which can lead to aggravation, or that we're not saying yes to someone doing something that might destroy somebody else's experiment, obviously. That happens via a Science Access Committee. And then in coming into BIFoR and starting to work on site, you sign up to obey the health and safety guidance that Chris looks after, but you also sign up to intellectual protocol and to a data protocol and data sharing agreement that protects your intellectual property, but is very much framed around open science, but with clear rules that the owners and originators of data are the people who we expect to use that data first. Across the country and externally, internationally, there's a growing network of collaborators, hopefully all mentioned here, but I wouldn't absolutely bet on it. And including those collaborators and all the people working on various projects in the University of Birmingham, we end up with an enormous list of people, and I wouldn't even say that this list is complete. And if you can't find if you have been working with us and you can't find your name here, my apologies. But I do want to say a great big thank you to everyone who has been contributing, because they've made a fantastic contribution. And I want to say thank you to the ECT. The ECT have provided us with funds to enhance or gap fill particularly important elements of the work that we do. By the nature of these talks, it can be that one is emphasizing the largeness of it and the expensiveness of it and so on. And yes, there is a massive investment in this facility from the university and a private philanthropist, and increasingly from UKRI. But that doesn't mean that there isn't a role for highly specific, highly targeted other interventions. And I was with the Woodland Trust and thanking them for making exactly that kind of targeted intervention recently. And I want to thank the ECT today for making those targeted interventions that, of course, leverage on a lot of other work that's happening here, but for that reason, they're extremely valuable to us, and I hope really valuable to you as a trust. So with that, I should be quiet and make some space for conversation.